This lecture is part of the Canadian Bioinformatics Workshop. It is freely available to share and do whatever you wish with the information as outlined in the Creative Commons license as we have here. So next part of the talk is going to be talking about enrichment maps, but specific use cases of enrichment map. Now we briefly mentioned what an enrichment map was and how we can translate our enrichment results into an enrichment map, but there are different ways we can go about using enrichment map. So hopefully by the end of this lecture, you'll understand the different use cases there are of enrichment map and be able to summarize your enrichment map results with annotations using the annotation apps and a few other the features that are embedded in Cytoscape and enrichment map. So first use case scenario for an enrichment map is a single map use case, right? A single experiment. So the example here is simply a treatment with breast cancer cells with estrogen at 12 hours and 24 hours. And we have 3 time points for treated and untreated. And actually, the single use case is here actually only one individual time point, right? So this analysis was done using just gene ontology. And in order to create this analysis, we found the differentially expressed genes for a given time point. We ran GSCA using gene ontology, and we translated those enrichment results from GSCA into an enrichment map. And so what you can see here is a simple network where each node represents a pathway, and the connection between the nodes represent the relationship between those two pathways. Pathways that are outlined in red are upregulated, and pathways that are outlined in blue are downregulated. And in addition to the nodes, you'll also see additional annotations on this network. You'll see circles around subsets of nodes with some sort of summarization term. Now, firstly, I just want to comment on the fact that this network was made a long time ago. It was one of the original papers of the enrichment map and when we developed it. And all of these annotations were done manually. But basically, someone came in, looked at each one of these sets of pathways that are coming up and attributed a thematic phrase associated with that cluster, right? We already know that from network analysis that finding clusters is something that we can do within the network software. So each one of these clusters are computed as nodes that are highly interconnected. The size of the node here is indicative of how big the gene set is. But basically, we've gone from a tabular set of pathways and generated a summarized view of all the different pathways that are differentially regulated at this given time point. You can see that there's a lot more upregulated gene sets than there are downregulated gene sets, and a lot of pathways relating to cell cycle and DNA metabolism associated with this estrogen-stimulated cells. You can zoom in on each one of these clusters if you're interested in seeing the actual names of the pathways associated with. So over here, I've zoomed in on the microtubule cytoskeleton, and as you can see, each one of the gene sets associated with this theme represents microtubule processing of some sort. Right? So you can zoom in or out and understand what's happening on an individual level. Now, another use case is the two case or the comparison of two different data sets, right? So over here, instead of just using one of the time points, I'm actually going to use both of the time points. So for each one of these time points, we've generated a different ranked list, and GSCA has been run on each one of these time points using gene ontology, but now we're going to visualize both of these results together. And this actually gives us a whole lot of other features of enrichment map that I can kind of highlight. Okay, so it's the exact same network, but what you have to notice here is that now there are actually 2 colors represented. So the inside of the node is actually the first time point, the 12-hour mark, and the outside of the node is actually 24. So we're basically overlaying 2 pieces of information on the exact same node. And what this kind of highlights specifically are pathways that might be on at 12 weeks and off at 24 weeks or vice versa, or just highlighting the difference, meaning that you can see down here this ubiquitin-dependent protein degradation, there's a clear difference between what's happening at 12 weeks and what's happening at 24 weeks. In the inside, they have no enrichment, but by 24 weeks, they do have enrichment. So this is, I guess, like the basic use case where you have two different time points or two different analyses that you want to look at. Now, also within the enrichment map, you can select individual nodes to see what's happening on the gene level. So let's say there's a particular area of the network that's of interest to you. You can click on a node or a set of nodes, and all of a sudden you can see what's actually happening on the gene level. So it enables you to have an overview of all the different pathways that are fluctuating, but at the same time, you can drill down into the details of what is causing this difference. A third use case. So you have your network and you've done your pathway network analysis, but you have some other piece of information that you want to get out of the network. Let's say you have a microRNA that is differentially regulated and you want to see which pathways the targets of this MER potentially could be part of. So there's something called a query set analysis. So a query set analysis or a post analysis. And here what you can do is you take another set of genes. It also could be drug targets. It could be specific disease genes. It could be anything. And you overlay this information on top of the network. And you ask the question, okay, where are the genes for this subset of genes found in my network? And so over here, I'm just showing an example where we had differentially downregulated pathways in a given sample that had a overexpression of a given MER. This was actually MER 125A. And then on top of this network, we actually highlighted where the targets of that MER are located. Now, I just want to comment, this is actually only a subset of the entire network. We took all of the pathways and groups of pathways that had a target of the MER, and we made a subnetwork. This isn't the whole network. It doesn't interact with the whole network, but this is just like a subview of it. The last use case is the multi-data set analysis. So there's no limit to the number of data sets you can input into EnrichmentMap. You can have 10, you can have 20, it doesn't matter. You can use it to compare pathway results across multiple conditions upon between multiple diseases, between multiple cancers, whatever it is. So over here, again, each node is a pathway, but it's colored according to a specific cancer in this case, right? So what you can see is there are specific pathways or specific themes that are more prevalent in certain types of the disease in this network. So this is an example of how you can use it for multiple data sets. Now, for each one of the colors that you see here, you actually have to run you have to have a separate rank list or a separate thresholded list that you run through gProfiler or you run through GSCA, and you're generating enrichment results for multiple analyses. This is kind of where, I guess, automation comes in. You could do this and you can run each analysis, but it becomes very useful to learn how to automate it when you're dealing with 10, 20 different data sets that you want to compare. So a few of the EnrichmentMap features, right? So when we were in Cytoscape, we were talking about the different panels, right? So I mentioned that a lot of apps will have some sort of input panel and they'll put it in the control panel of Cytoscape. So over here, I'm just showing you the control panel of EnrichmentMap, where you're able to play around with some of these like slider bars. You can create an EnrichmentMap and then decide that you want to play around with the p-values. So if you get a network that is highly interconnected, you can actually lower the p-value directly from the interface and see how the number of nodes you have in your network changes. And the same goes for the connectivity threshold or the overlap. If your network is too interconnected, you can actually lower that threshold to try and clean up your network a little bit. Also, within the table browser or the table panel of Cytoscape, EnrichmentMap adds a heat map, which I showed briefly before. When you click on an individual node or a subset of node, you can actually get the expression of all the genes associated with your selection. You'll see it a little bit more in detail here, where this is the heat map panel, if you select an individual node, only if you select an individual node that is the result of GSCA, you can also see which genes are part of the leading edge. As we mentioned when we discussed the GSCA, algorithm, that there's a subset of genes when you look at that enrichment curve that are actually causing the enrichment. So it's all recorded in the output files from GSCA, and EnrichmentMap uses that and can actually show you which genes were part of that leading edge. Now, again, that's only if you select on an individual node. If you select a cluster of nodes, you're not going to see the leading edge. You'll still see the genes associated with the subset that you've selected. It's just you won't see this highlighted region that is indicative of the leading edge. Okay, so one other thing that we've seen in quite a few of these pictures are these annotations, right? So you have your enrichment map, and then on top of your enrichment map, it draws a circle around stuff that's interconnected and it types a little word. Now, this actually is an additional app within Cytoscape that is doing this, and it uses two other apps in Cytoscape. So it uses Cluster Maker to calculate the clusters, and it uses word cloud to try and guess what the best annotation is for that cluster. Now, it's all done automatically, and I need to just remark that it's not set in stone. If you don't like a annotation associated with your cluster, you don't have to keep it. You are the expert on the biology, and it's literally doing this automatically, meaning it's counting the words in the network, and it's normalizing the number of words it has in your cluster to try and guess the best annotation. Truthfully, this is done very simplistically, and hopefully as a new feature, we will be able to integrate more things like ChatGPT to better get annotations from the names of the nodes, something that is actively being thought of. But for now, all it does is it counts the words and it generates a key for it. So what does that auto-annotate do? It clusters the network. There's lots of different clustering algorithms that you can use. If you have your own clustering, you can also supply those. But by default, it uses an MCL clustering. For each cluster, it finds the frequent words in the node labels, and it selects 3 of the top words and creates the label. You can edit these nodes. You can edit these labels. You can change the label to whatever you want. It is not set in stone. It's just kind of giving you its best guess, and it's something to start from. So auto-annotate will generate a list of terms for each one of your clusters. You can change the way those annotations look by changing the labels. You can tighten them, making them smaller. You can make them a square. There's lots of different features you have to use when you play around with auto-annotate. You can right-click on any one of these and rename any one of those clusters. Another, important feature is that you can also collapse clusters. And you can, if you have a massive network, you can create what's called a summary network, where it takes every single one of the clusters and it collapses them and uses the name of that cluster as the name of this collapsed node. So what you're seeing here is actually, we had a very large network. You can kind of see it over here, very, very, very large network. And I'm just showing you a small subsection of that network which has been collapsed. It basically gives you a summarization of all of the enrichments that are coming back for your given analysis. Okay, so that's just a basic overview of EnrichmentMap. You're going to get a chance now to try out EnrichmentMap and load in the gprofiler results and the GSCA results that we did from Lab 2 and try out all the EnrichmentMap features.